Hey, good morning, everyone. My name is Srdjan Achimovic. I'm a plant pathologist at Virginia Tech here at AREC in Winchester. Thank you so much for getting time to attend the webinar on some of the experiences that I had in using RIMpro. So today I'm going to be showing you information about the RIMpro apple scab model, how to interpret it, and also the fire blight model, and just show you some basic things about peach brown rot prediction model as well. So hopefully this is going to allow you to get more information to incorporate this model into your spray decisions. And this would be the only tool that I personally see as the most important one to help you deal with unpredictable weather that is a promise of the climate change. So the first model that has been incorporated in RIMpro is about the apple scab pathogen Venturi equalis. So what you see here is the life cycle of this pathogen. You can see that the pathogen overwinters in the leaf litter from previous year. So dead leaves where the fungus is capable of forming its sexual structures. And you can see them here as pear fruit-like bodies that have an opening at the top. And this is where the pathogen overwinters, and in the spring, these initial perithecia get differentiated, and they get ascospores in each of these sacs. And then the ascospores in the spring, with enough rain and light, they eject ascospores into the air, and they are carried by air current, landing on leaves, on green tissue or green fruit. So these ascospores that you can see here that are bullet-like, they actually require being in the droplet of water to infect. So once the infection occurs and they penetrate through the basically the cuticle of the leaf or fruit, the pathogen then goes through the incubation period and forms conidia that you can see here as a candle-like light spores, or conidia asexual spores that basically protrude through the skin or the waxy cuticle of the fruit. And then if we get to the stage that we can see the symptoms in these conidia, it becomes much more complicated, and these conidia are not as mobile as ascospores, but they can cause multiple cycles of infections because there can be multiple generations of these conidia throughout the season. So if the infections occur on the fruit, the yield is lost or is severely reduced. But then also the infections on the spur leaves early in the spring lead to damage that prevents the leaves to feed the fruit. And so these infections early in the spring have to be prevented. And throughout the whole life cycle of the pathogen, all of these stages of formation of perithecia and ejection of ascospores and then germination, they have all been investigated pretty well, and meaning that the specific ecological factors for each of these stages has been very well studied and then incorporated into RIMpro. So the symptoms on the leaves you can see here is olive green spots that are visible here. Eventually, these olive green spots become full with conidia, and they become dark or fuzzy black velvety spots. And these early spring infections allow inoculum to spread further on other leaves and fruit. But then in the fall, if the protection has not been continued, in the fall you can have sheet scab that develops on these leaves. And the sheet scab is specifically problematic because in these leaves, for these infections that happen late in the summer, this is where the perithecia are going to be abundant in these leaves when the leaves drop. If we get the infections on the fruit and on the petioles of the fruit can drop and be severely deformed. But the later infections, when they occur on a golf ball sized fruit, they can lead to cracks on the fruit and complete loss of the crop, as you can see here. So right now, about this time of the year, one of the best practices you can apply, so as long you don't have the leaf, so as long you don't have the snow above the leaf litter, is to try to eliminate these fallen leaves that allow pathogen to overwinter by applying urea or lime. This will help you to reduce the ascospore dose for about 50 to 70%. So this is, of course, just one in many management practices you can apply, because even these remaining 50% or 30% is enough to cause a lot of infections. But at least by applying urea or lime and reducing the ascospore dose, you are doing a better service to allow your fungicides to be more effective. Usually the rate that we apply at this time would be about 40 pounds of urea per acre in at least 100 gallons of water. And the way how applying urea would be done is by deflecting or using just the lower nozzles on your sprayer, on the air blast sprayer. And if you are not using urea because you're maybe an organic grower, then the only option is the dolomitic lime, using about 2.5 tons per acre, which is much more intensive to apply, but you can use spreaders that would be recommended to spread lime anyhow in your lawn. You can use those to apply it. In the case of lime, it increases the pH or basicity of your which is opposite from acidity, increases the pH of the soil, and specifically on the surface, which promotes the microbial activity. So those microbes are there, promoted to try to degrade the leaf litter and then prevent in that way overwintering of the apple scab. So in case of the urea actually is also a food source for these microorganisms and also the snails like fauna in your orchard. So they also allow the faster degradation of the leaf litter. If you have a flail mower, before you apply the urea or lime, you can actually use the flail mower to shred the leaves into smaller pieces, because then the degradation by the microorganisms or fauna is going to be much faster. So some people decide to rake the leaves from under the trees, put them in a strip below the trees or below the mid rows, and then use the mower, flail mower in a scalp mode to basically shred the leaves. Or sometimes they remove completely the piles out, and using the different methods. So the scalping mode of the flail mower is really good because it shreds the leaves in small pieces. In general, what will help you also is to prune the trees so that you can have a good air circulation. So trying to keep the density of the crown low is great, because that also allows better coverage with your fungicides. So this method also helps with Marshinian leaf blotch as well. So using the urea and lime is not going to also reduce the amount of inoculum of apple scab. It's going to also reduce the inoculum from Marshinian leaf blotch. So let's now talk more about the RIMpro. So in 2016, when I started working at Cornell, this model started being intensively used in the United States. And it was developed primarily for organic producers of apple in Europe. The headquarters of the company is in Netherlands. So we started the partnership there. And then when I moved to Virginia Tech, I started the partnership with growers to try to learn how to use this model here in Virginia. So for like about 9 years now, I have been involved in looking at the RIMpro, and we are continuing this partnership in trying to outsource this tool to the growers. And you can see here how this model looks like. And what we're going to do today is we're going to try to cover what does this model give you that is different from other models. So this partnership I mentioned to you before had a large number of stations, weather stations, that were on the growers' orchards that you can see on the map to the right in New York, Vermont, and New Jersey. And we connected all these stations to RIMpro using the NUA network that is hosted at Cornell. And all these all the information from the models was then spilled over into emails. So basically, when you subscribed to RIMpro, the data that you get is your own personal property. You can't share it publicly, but you can designate a group that connects all these stations together, and then you can have consultants form this group and then work with you one-on-one or through emails to interpret these models, which is not considered public sharing. So from 2016 to 2020, you can see here a number of blogs that I published. Actually, not the blogs. Actually, those were emails that I published. For the people who were subscribed to RIMpro. But then, I also used this information to kind of refine better the blogs that I would publish also with announcing to the growers what's going to happen in the next 7 to 10 days. But specific dates and specific timings when infections are going to occur cannot be shared because that's your private data. It can only be consulted or interpreted by me through the emails. We also, through these blogs, developed and actually sent information about when we saw the first symptoms of the apple scab, and then we also would deliver efficacy trial results. So we would use the model to run the efficacy trials as well. What you can see here is the microscopic examination of the leaves that I mentioned to you in the leaf litter. These are the ascospores visible in the center of the image of the slide. You can see the ascospores that are visible under the microscope, and then on the right side you can see the conidia, how they look like. If the infections occur in the leaves and you get the formation of conidia, then the situation is much more complicated to manage. So they are definitely different. So ascospores are much more airborne, whereas the conidia are mostly rain splashed and they don't move much, in distance. So in general, the ascospore maturation is predicted, and it starts when the relative humidity in the air is about 70% and above. So that speed and the amount of the ascospores that mature is usually assumed to be proportional to the accumulated temperatures above 32 Fahrenheit. So it starts with these temperatures accumulating above 32 Fahrenheit. And many times when we use the spore trap to see when the ascospores are actually, if we can trap them and find them under the microscope, about 50 to 60 of those spores is the time when we start to be concerned, and we basically report that as a biofix when we get about 50, 60 of them, and we call it an economic threshold, from which we start calling that a biofix. So if we detect that in the vacuum spore tower, then we would go and put that data as a biofix in RIMpro. So the way how we do that is we would put the leaf litter from previous year. We would find really good leaves, about 60 to 50 of them. We put this nice plastic tarp and put them there and put the rocks so that we can actually take the batch of leaves every second or third day, or maybe weekly once, and then run the Scab spore tower by putting those leaves here on the top. Vacuum would be applied in the lower part, and then we would have down here 3 or 4 glass slides for microscope that would have Vaseline on them. And then you can see here, we would collect the spores that would be ejected from the leaves, and we can actually see under the microscope how many of them we can see them. And we would count them. Sometimes, we would shoot through this 50 to 60 spores threshold immediately within a day, but that's why the spore trapping should be done almost every second day or every day, so that we can actually not go over this threshold of 50 to 60. So, and then we would use the date when we get this 50 to 60 spores, we would use that date as the biofix to run the model. The other biofix would be to use the green tip date, which is about 50% of the floral buds being at green tip. And the way how you would determine the 50%, have you reached it or not, is by actually doing the counts. We do this on the most early cultivar in the orchard that is susceptible to scab, so and specific on your farm. So we would take about a sample, go through the orchard and do a transect walking around, and we would sample about 100 buds on about 10 trees. And if we get about 50 of them out of 100 that we sampled, we would call that a 50% green tip. And then that date would be also a biofix you can incorporate into the RIMpro. So both of the biofixes really allow the model to work much more accurately than just using one of the biofixes. But in the case when there's a location at which you don't have access to spore trapping, you can definitely use the green tip as a date. And there, the RIMpro moderators told me that there's not much difference, but we have seen much better working of the model if we use both of these biofixes. So we're going to hope and try to collect the biofix, which is the ascospore release. We're going to try to do that here in Winchester this season. And then also what is important is that this spore bank in these leaves can also be depleted. And we call that we call that the primary scab season end when the spore bank inside gets depleted below the 5% of the mature spores. And we're going to talk about that more later. So this is the output, as I mentioned to you, of the RIMpro model. And it does seem a little bit busy, but I'll go slowly through it and explain to you what is it that you can see here in this graph. So the most interesting thing, of course, is that if you see the letter A, there's this blue line that is vertically present here, and what this blue line denotes is the separation of the graph in two sections. On the left side of the graph, you can see the historical weather data that was collected from your station. And those are actually real ground truth data that shaped the model in what actually happened on your farm and how that affected the pathogen to infect. On the right side of this blue line is the prediction of the weather forecast, and then how the model looks like when this predicted weather is incorporated into it. So we would normally not rely on any weather forecast that is beyond 48 hours. And the reason being is because the weather forecasts are really unstable beyond that time. And sometimes when you would see like a big infection here that you can see here, this red line, it denotes infection. This actually, because it's farther in time than 48 hours, many things can change and this huge infection might completely disappear by the time we actually get to that day. So we only stick to about 48 hours forecast that we can see in advance. So the B here that you can see here are these white humps. They're like a white camel humps that you can see here. And what this denotes here, as you can see in the graph, it says germinating spores. So these white camel humps show you a cumulative number of the ascospores of apple scab fungus that germinate. And you can see how that whole germination occurs through time. And also what is important is that the height up until which this white camel humps go gives you actually the number of these spores that can germinate. And those are the cumulative numbers across multiple hours when the wetting lasts. And for you to understand how much of the spores accumulated, you go to the height of this camel peak, and then you read the amount of the discharge you can see here on this scale, Y scale, Y-axis on the right side of the graph that says discharge on it. So besides that, what happens is if many times when the spores can actually germinate, like in this case here, they're released, they can germinate, and we see the cumulative amount of spores that germinate. But many times the infection does not happen because maybe the temperatures are low, and it's generally cold for those infections to occur, even though the spores germinate. So when we see these white humps without the red line that is followed after that, like in this case here, we do see a line followed after. The infection in this case here, when we don't have the red line with it, it indicates to us that yes, there were conditions for spores to germinate, but there were no conditions to continue infection, and these spores failed to establish the infection. So in the case when you use this model to zoom in, and so the letter C here, you can see here that I'm pointing you to these small, kind of like a very narrow lines that you can see here. And when we actually zoom into this model, using the zoom tool, like a magnifying tool, you can see here these yellow bars that indicate to you the ejected spores. So with each hour, you can see the injected spores. And so that's just one way to see how much spores is ejected. And you can read that again on the right side of this y-axis on the right side of the graph. So that information is there for you, so you can see how much, okay? But then when we go back here, we see this cumulative amount of spores that germinated. Now we want to know, okay, how many of them caused the infection? In this case, none of them caused infection, and applying the fungicide would be not warranted. But in the case of this red line forming after that, and this red line actually is called the RIM value, or relative infection measure, in short, that shows you what is the percentage of the ascospores that actually, from these cumulative number of that germinated, how many of them actually caused the infection. So, and the height upon which this red line goes is red on the y-axis on the left side, that is called RIM infection value. And the way how you can actually determine the percentage of the ascospores is by taking this RIM value, and let's say in this case here, for the April 29th, we got to about maybe 350. And that's where the height is for this red line. If you divide that, 350 by 100, you would get 3.5. You would get the 3.5. Oh, I'm sorry. Yeah. So you would get about, when you divide it by 100, you would get the percentage of the ascospores that caused the infection. So in this case, you get about 35% of the ascospores that caused the infection. Actually, it's a 3.5%. I'm sorry. So that tells you that yes, this was a high infection happening here, and in some cases here, when you can see here on the May 13th on the far right, this infection went above 1,000. So you would get about probably 12-13% of the ascospores causing the infection. Then finally, what you also get here as an information is this orange field that is behind this red line, the red curved line. And the orange field is called, as you can see here, primary stroma. This is a designation where we know that the incubation is happening. So the ascospores penetrated into the subcuticular space of the leaf, and the incubation is occurring. So, and this period here shows you the orange field shows you the time up until which you have to apply the material that has a post-infection activity. So there's a window. Let's say if you didn't apply fungicide before the infection, you can have still more time to apply systemic fungicide that will allow post-infection control of the infection that is incubating. So the other information that this model incorporates is the graph in the middle here that you can see, which has a dark red like a field, and then it has a light red field above it, and that's actually your spore bank. So how many ascospores are still remaining in the leaf, being immature, which is the dark red, and then the light red shows you the mature ascospores in the leaf bank. And basically, so with each rain, these mature ones would be released. So when you get to the point that this spore bank gets below 5%, we would call that primary Scab season end. And finally, at the bottom, you see here light blue and dark blue like bars that indicate to you, the dark blue bars show you the length of the rain, how long it lasted, and the light blue bars show you the length of the leaf wetness after the rain was done, and sometimes it's just dew. So all this information you can find in on my website. You can get an article that has all this information there. Also, I'll be sharing the presentation today, and all this information that I mentioned to you is also written in this presentation. Okay, so I've explained to you what these parts of the graph mean from A to D and everything, but the one thing that I wanted to pay more attention to is to talk about for which specific RIM values you should spray the fungicides. So if your orchard did not have scab last year, usually what we would say is that that's a scab clean orchard. And in this case, you would try to next year, after the year when you did not have scab, if you have rain events that generate the RIM value or the relative infection measure of 300 and unless, you probably will not have any economic consequences from that So anything below 300 rim value, if your orchard was, was clean last year, you would not need to apply fungicides. However, if you had any scab, any small amount or even larger amount, of course, so meaning that your now ascospore dose is higher that you carry over into the next season, you would have to apply fungicides for any wetting periods that generate a rim value that is greater than 100. So because you have much higher ascospore dose. So that will be something that we have done before, and it works pretty well. Finally, again, I'm continuing here to talk about each of these sections of the graph that I mentioned to you verbally, but you can read through it later on by getting this presentation. And so I'll just continue talking about the advantages of the model. So in discussing this model with Vincent Filion, okay, so by talking to Vincent Filion from IRDA in Canada, who is one of the people who is involved in developing the RIMpro model, RIMpro actually incorporates in it much more biological aspects of the fungus epidemiology, and also because the RIMpro incorporates other disease models, all of them incorporate many aspects of the epidemiology than what was the case maybe 30 years ago. And they call this model and all the models there as more dynamic and not static. So what does that really mean? So if you have a first ascospore of the apple scab fungus that lands on the leaf, and this, that first spore and maybe the last spore at the wetting event that landed on the leaf, they actually have different destinies because they have different conditions. So the modeling, basically accounts for all of those specific conditions that first spore has or the last spore has to be able to germinate. So let's say that maybe the first spore landed in the evening and the last one, landed, the next morning through the whole wetting event. What happens in the NUA model for Scab, the model sees only one rain event, and this model is static. It uses just, it basically starts counting the infection hours when the rain started, and and basically what happens is, if the rain started during the night, then basically the infection should not happen. But if the model starts counting the infection hours in the morning because the light occurs, so this is just very like a yes or no or digital response. So it doesn't do anything else. Unlike these models, that are static in that sense, so they basically don't count the infection happening during the night, or do count the infection when the morning starts because ascospores require light to infect. So in contrast to the static model, RIMpro actually dynamically accounts for all those conditions because yes, there's a very small amount of ascospores that do get released during the rain in the evening or during the night. So that low ejection is definitely reflected in RIMpro. It accounts for those being there, and then it keeps track of these spores and their behavior. And then when the temperature changes in the morning and the day arrives, there's light. RIMpro knows how to accelerate the ejection of spores because they have actually incorporated inside all the biological requirements of these spores that now are ejected in huge amounts because the light is present. So, and then the model is also dynamic because it follows all groups of spores that are ejected, and they're through the time at different conditions. They follow these groups of spores and their life stories from the beginning until the end of the wetting event, from spore maturation, ejection, and infection, which is definitely different from the static models like NUA is. Okay. So I'm not the only one who has been
Matthew.
I'm not the only one who has been involved
Morning.
in using this model. So John Clements at
Still
UMass.
recovering from my bronchitis. What about you?
John, I think we can hear you, so maybe you should mute yourself.
It's not good. Where are you?
I'm in the office today.
Oh,
Yeah. How was your trip?
is there anything else there?
John, if this is John Clements, can you please mute?
Here? Not really.
So John is using also this model and also incorporates some of these decisions from this model into his blogs. So he also has been working on this for several years, and has been using it both with using the actual data from the weather stations, but also using the data from virtual weather stations. So using this forecast from Norway that is globally used. So it's not only my lab that has been involved in using this model. So also the group at Dan Cooley's lab at UMass has looked at the model and did some validation work. So you can try to find this article online. It's freely available to download, and you can read through it. But what they have done in this paper is they have used the green tip only as the biofix for their work. Whereas in my case, in my article that I did, we've used both biofixes using the green tip, which is 50% fruit buds at green tip, and also we used the ascospore trapping to kind of increase the accuracy of the model. So what we found was we generally looked at this over like 3 years, is that many times what we saw is that early in the season you can avoid 1 to 3 applications of fungicides, and we looked at this in 2 years, and that will specifically be early in the spring. And if you would avoid these 1 to 2, and of course that will be in the cases where even though the spores germinate, they don't have the conditions to finish the infection. 1 or 2 additional sprays also we found were needed to control the scab at the end of the season. So the model would predict that there's more applications needed at the end, close to the primary scab season end. And if you really looked at the savings you can get from omitting these 1 to 3 fungicide applications early in the spring, it will be about a $300,000 to $900,000 for about 12,000 acres that we covered in Eastern New York. So let's just see now what those infections would be. If you looked at the NUA model, all these ascospore releases that you would see here in germination, cumulative germination, would be infection periods for which you would need to apply fungicides. Whereas the RIMpro would tell you that these white humps, yes, there were ascospore released from the leaves, but the conditions were not conducive for these germinating spores to finish the infection. So in these cases, you would have 1, 2, 3, or maybe even 4 fungicide applications that you would not need to do. So this was in 2016. Then we have about maybe 1 or 2 applications early in the spring of the 2017, up until you get to a really big infection here that you need to apply fungicides. In 2018, there will be only maybe 1 omitted application here that is not necessary before you get a first big infection period. In 2019, you will probably have 2 or 3 applications that you don't need to apply here because the conditions are not there to finish the infection before you get to a big, large first infection period here. So let's just now go and guide you through the process of how you would go ahead and decide if you want to use the RIMpro. How would you go and use the RIMpro? So the first way to allow your station, if you have your neighbors or your station, so you don't have to own the station, you can use the one that is next to you, okay? Because all the data from these weather stations, rain-wise weather stations, is flowing to the NUA's website at Cornell University. So if you Google new RIMpro license, okay, you will get the second hit here, and you would get this data license request. So you have to basically pay a small fee to allow this data to flow to the RIMpro model from these stations. Okay, so this is what you will get after you reach and click that link there to get the new license. So, and then when you click on go to the new RIMpro license portal, you get to the NUA station RIMpro data feeds, and you click continue. And then you put your name, your email, phone number, and your zip code, and then you basically choose a state. And when you click on this one, submit request, then Dan Olmsted from NUA at Cornell is going to get a request to allow this data to flow from your station to the RIMpro. And it's going to take him about maybe 3 to 7 days to get you a data link on your email that is necessary for RIMpro to actually function. So this is the number of stations that I currently have in Virginia. Okay, there's about 7 of them right now. And this is the way how the map looks like when you actually log into the account, when you pay the subscription. So, and the first most important thing is when you get that data file link, just email it to me and send me your password and your username for subscription for your RIMpro, and then I will connect your station to the RIMpro. And then I will probably call you or text you or email you to work with you to set your biofix. Specifically, I would ask you, have you reached green tip on your earliest cultivar that is susceptible to scab? Let's go get that date, and we would put it inside of the model. So I need your password and your login as a consultant so that I can actually put that data in, because it's not that easy and it's pretty tricky to get all these dates incorporated into the model. You know, the RIMpro, you know, has been telling me that both biofixes, the green tip and the first ascospore release, are equally accurate. So, and I have been using the 50% floral buds at green tip as a biofix in the last 3 years, and I haven't seen much difference myself. So now let's go and look at the way how the model, you know, opens. So I'm going to just go and share my screen and just show you how, you know, you can log in. If you create your account in the RIMpro and you open this model, you know, there's you know, the map that you get with all the stations that are grouped inside, and you can basically go and click on the specific location that you're interested in, with the default model being opened always being apple scab. So if you click right now on a station of your interest, for instance, in this case, it's Quickspurt, you know, the model is going to start using the data from the link that Cornell provided you and start, you know, generating the actual prediction of a disease. And it takes some time sometimes. There's a lag because all that data needs to go to the server in Netherlands and then come back to you. And apparently it seems that it's not working this morning. And sometimes, okay, so here's one for the data from last year. You can see how model looks like. And I mentioned to you before that there's many tools you can use on this graph to, you know, look at it. First, you can actually go and click on a drag graph like this, and you can actually see, you know, what's happening at different dates across. You can click here to zoom in and look at the actual specific events. And basically hone in more on specific events that you're interested in to look at. Right now, it doesn't seem to be working. I'm not sure why, but you know, it has to be subscribed to be able to function. So here again, let's use the zoom in tool. Okay, so now we can actually go and actually look more for each infection period. And then you can see these specific small yellow bars that I mentioned to you before that shows ascospore release from the leaves per hour. And then you can see cumulative germination is white hump. Then you see the actual infection happening is this red line. Okay. You can also zoom out and go back into the mode when, you know, you're looking at like a larger time scale. Okay. So on the left side here, you have many options for each locations. Okay. So my stations that I'm using are the ones that are present here at the Winchester station. If you click on them, you know, will be able to kind of switch between the stations, but also, you know, within that group, I have many stations that are connected, and you can actually go and set up the parameters for each station. That's where, you know, you would go and get the biofix, you know, implemented there, so that you can actually change it. However, I find that pretty tricky to do for many people. So that's why I'm asking you to send me, if you do decide to subscribe to RIMpro, just email me your password and your username. You can trust me. I'm not going to do anything, bad to your RIMpro. I'm just trying to make it easier for you and provide you technical support to set up your stations and connect you to your your data file from your station to go and pick up the data so you can feed the model to work fine. Also, what you can do is if you go back and if you log in, you can also pull up different models. You can actually click here and you can choose different models that and for instance, in this specific case, you can pick up the Blight model, choose the location where you're in, and then pull up the visual for the actual Blight model. There's many other models you can use for grapes there, and also there's stone fruit models as well. But I'm going to try to pull up the model for Blight, because this is the before the season starts. Sometimes this doesn't work up until you pay your license. That's probably the reason why this is actually not working at this moment. So I've shown you that one of the stations here had the data from 2023, but right now it doesn't seem that it's working because these stations, the subscriptions have not been paid, most likely leading that to be the issue why we don't see that. But anyhow, what I'm going to do now, I'm going to go back to the presentation, and I'm going to continue talking about the this model. Okay. And so keep in mind, when you pay the subscription, they're very strict. When you subscribe, you own the access to the information, but you cannot share it publicly beyond that subscription. You can share it with me as a consultant, and I can help you interpret the model and teach you how to learn how to use the model. But then in general, they can shut you down if you start sharing publicly the data. So I can work with you in a group to help you how to use the model, and I provide technical support, and I can help you learn how to use and connect the model. So the next thing that I want to talk about is the conditions in Virginia. So this is the sample that I had in 2021. So I'll show you 3 years of data in Winchester, and you can see here that that we have about 3 to 4 major infections usually per season in Virginia, and maybe in some cases we have about 1, 2, or 3 false infection periods that would not warrant application of fungicides that you can see here. So this is how the scab season looked like in 2021 in Winchester. This is how it looked like in 2022. We had about 4 major infection periods. You can see here. And in this specific case, this infection here would not be warranted to apply fungicides. And we had about 4 infection periods that were major in 2023. So that's about just the differences. And for these 2 early infection periods, or false infection that I would call them, you would not need to apply fungicides all the way up until the 16th of April, when we would have the first major infection. Okay. So I'll switch now and talk about Blight. This is the life cycle of the disease, and I'll not linger too much on it because I want to focus on RIMpro. These are the symptoms of Blight, which of course lead you to lose the yield and the current year growth if you get shoot blight, but also if you allow the blossom blight and shoot blight to set in, you would get the pathogen to invade the wood and form cankers where the pathogen can survive. So these cankers can perpetuate disease in the future, because that's where the pathogen actually overwinters. But it can also lead to girdling of the tree or the central leader, but girdling of the actual central leader or the rootstock to kill the tree. And so that's why the Blight is very dangerous in high density orchards. And this is how the dead trees look like. In an orchard, you can see these purple dead trees that have cankers on the rootstock or on the central leader in a high density, super tall spindle orchard, whereas these other ones are normally senescing because they're still alive. So in high density orchard, the risk of Blight is much higher. This is how the NUA's mare blight model that is incorporated and freely accessible online looked like before the new update they made. So as of, I think, 2021, this is not the way how the model looks like. But anyhow, I wanted to show you here this mare blight model incorporated online was showing an infection here happening on the April 29th. And in the case like this, when you have the infection potential about 326 and there's a rain event happening, it's relatively easy to come here before this and apply your material before and kill this infection, or actually kill the bacteria that caused this infection, and be fine. But when what we saw in the last several years is that we would sometimes have 3 or 4 or 5 days of continuous infection happening. So the conditions are extremely conducive for infection to occur. So it becomes really mind-boggling. What do you do in these cases? These here are the requirements for the infection to occur: open flowers, accumulated heat units, which would be your infection potential value. So this accumulated heat units allow the pathogen to grow its populations on the flowers to reach the threshold of about 1 million cells. And then you have to have a wetting event, either dew or water from your spray or rain, to allow these bacteria on the stigma of the flower to get washed down into the nectar glands. And the average temperature should be about 60 Fahrenheit. And the cutoff point for us when we start getting worried is about 100 for EIP and above. So when you have a situation like this, May 3rd, 4th, and 5th, when the infection potential value and the rain was indicating to us there's going to be infection, people get confused how many times they should apply materials or streptomycin to control this disease parenterally. And I'll show you 2 cases here for 2 application times. So you can see here that the model was predicting infection, as you can see here, on the May 3rd, 4th, and 5th. So now I put here the date of streptomycin application being May 3rd. And if you apply the material on the May 3rd, before the infection, you would be able to control the infections of the 4th and 5th. And because the model resets and shows you how the EIP looks like after you applied on the May 3rd. But if you decided to spray on the May 4th, the model would tell you that the infection still occurred on the May 3rd. So this is very important for you to understand that sometimes when the conditions for infection are extremely conducive, you have to apply more streptomycin applications to be able to control the infection properly. Okay, so this is the output for the RIMpro model. This is giving you much more information than just what I showed you in the Mary Blight model online for NUA. It's a little bit busy, but let's go from the bottom of the graph where you can see actual rain events as dark blue, and you can see the light blue color that shows leaf wetness at the bottom. Then above that, there's this graph that is called the epiphytic population CFU that shows these curved lines. They're either red or black. So each of these curved lines at the bottom graph is actually a group of flowers that supports the population of bacteria. So and they start at irregular intervals, okay? And the way how the model functions is when the temperature gets to 59 Fahrenheit, the bees become active, and the model starts accumulating basically the amount of bacteria, looking at the amount of bacteria that will be inoculated by the bees on the flower. And that's why you see these lines started at different intervals. At any time when this 59 Fahrenheit kicks in, there's a potential that the populations are going to grow. Some of these lines are red, some of them are black. The red ones are the ones that are actually the ones that when they enter this orange field, they can cause the infection if the rain occurs. And that's what you can see here above these red lines, curved red lines. You can see that in the middle graph, it shows the incidence or infection threshold. So these small red bars indicate you when they go over this dashed line that the infection has occurred on the flowers. And then above that, you see this other graph that has a green big bar above it that shows you how much time it is necessary for this infection that occurred to incubate and then lead to formation of symptoms that you can see. So this is what these other red lines up here are indicating to you how long it is necessary for this infection that occurred to lead to formation of symptoms. And some of the lines are black. So in this specific case, these lines are black because there was no rain event that followed this black line. So that's why they're not showing that as a red, the population that led to infection. So why is this important to you? It is important because it pays really a good deal of money to know when the population that is going to cause the infection has started. And if you apply your streptomycin at any time during this curved line, is actually progressing, you would intersect this population with your material and kill it. And this is very important in large acreage orchards where you need more time to cover. So you have this window between here and here to apply your material to kind of cover your orchard and prevent the infections to occur. Of course, under the assumption that the weather forecast is reliable and it's accurate. So if you have large acreage, this model can actually help you by giving you the insight of the biological information of the pathogen, how the populations are growing, to actually control this disease before the actual infection occurs. So basically be more preventive than just applying it 24 hours before the infection occurs. Okay, so what I just mentioned to you here is also written in this presentation by text. So you can read through it. I'm not going to go again through it here. So I'm just going to leave it here for you to read more later. So in general, this is the same thing what I said when I was looking at the graph. This is the way how the NUA's mare blight model that is incorporated into the NUA looks right now. And you have the cougar blight incorporated here, and you also have the infection potential EIP value on the right. And by and large, because this mare blight model EAP value was developed by Paul Steiner at University of Maryland, we mostly rely on the East Coast, on using this model. They couldn't call it mare blight because of the copyright laws, but it is that model incorporated into the NUA here. So it's a little bit more simple to look at it now. It has dark red lines here that basically indicated to you that on these days, on May 4th and 5th, the infection is going to occur when you have a dark red line with EIP value of 100 and above. And if the rain occurs on that day, this bar is going to change from orange to dark red. And for those, you would have to apply the materials. What we have seen is that the mare blight model on NUA's website largely aligns well with the model on RIMpro. So I'll show you here now for the data of 2022. You can see here that in Winchester, we had 3 infection periods. I'm just showing you here how the season looks like. You can see here that there's 3 times when these bars went above the stashed line. And you can see 3 populations that led to formation of enough bacteria to infect. Then, I wanted to show you a southern location here in Rossburg. You can see that they had many, many infection periods. So this also largely aligned with mare blight. And what I'm trying to show you with this is that it became in the last 4 or 5 years much more complicated to manage fire blight because we have highly conducive conditions for fire blight. They're abnormally conducive. I mean, really, really conducive for fire blight. In these cases, it becomes really difficult to decide when do you want to apply your materials. And here's the 2023. In Winchester, we had 5 infection periods for fire blight. We did the trial this year, and these infections, we would apply certain materials at these times, but I can tell you that the more north you go, potentially this frequency of infection periods is less, but the more south you are in latitude, you can have more infection periods, which becomes a huge problem, especially for growers who have cider varieties that have a prolonged bloom. So having a model like this is going to allow you to time better your streptomycin against the fire blight. So let's go and look at just an example of a trial that I did last year. So I've tested here some experimental materials that you can see here in green, but I want to focus you on the number 3, which is the firewall. So number 3 is can you please not use the laser on my presentation? Whoever is using it. So you can see here that there is treatment number 3 is the application of fireline and firewall with regulate. Whoever is using the laser, can you please stop doing that? I would appreciate it. Okay, so we have a firewall and regulate. This is what I'm interested to show you, the application of streptomycin and oxytetracycline. So this is basically the time So this is the 2023, when I showed you that we had the 5 infection periods, we decided to apply inoculum, so to inoculate this trial on 14th of April when we were in bloom. So we would basically set up this experiment inoculating with Fire Blight. So I want to go back here and just want to show you that we applied the Fireline and Firewall. In this case, the number 3 treatment was on 60 to 70% bloom. Okay, so just before the infection or the inoculation on the 14th April. So just the day before. And so again, I'm showing you here how it looked like, and then also that this also largely aligned well with the mare blight in NUA. And I can show you here that the treatment number 3 actually has control on blossom blight about 20%, which is the blue bar here. So it's known that the application of antibiotics is going to allow you control of the disease of about 20, I would probably say 10 to, allows the infection to be about 10 to 20%, but in general, it has a good control of the disease in comparison to the control. So applying your material before the infection is crucial, and it gives you much higher efficacy of control. If, God forbid, you do get fire blight, okay, now you have to go and try to do pruning and then continue spraying certain materials that are allowed. And here I'm going to show you 2 scenarios late in the spring and early summer. Usually what we understand is that when you see the first symptoms is that, and usually that's going to be about 90 to 100-degree days with a base of 55 after the infection event when the symptoms are going to occur. So you have to keep on scouting to see whether you see more symptoms. And if you see a low number of symptoms, about 5 to 15 strikes per tree, you should probably go out there and rapidly prune and definitely sanitize your tools. You know, cutting these strikes about 18 to 24 inches below the visible symptom edge, because there's probably a chance that the pathogen has moved, invaded through the tissue into the wood, way beyond what you see as the edge of fire blight. And the idea with this low infection amount that you see about 5 to 15 strikes is that you want to prevent the further spread of inoculum on other trees. And in general, what I'm trying to say is that when you cut these, strikes, you can just drop them in the middle, let them dry, because moving the brush that you just cut out is going to allow the pathogen to spread, especially if you have dew on the grass. So the pathogen can find the way to be spread around that way. So that's why we are recommending to just drop the cuttings in the middle, let them dry, and then come later on about maybe 2-3 weeks to remove it. Please mute yourself if you are using the Zoom. I would appreciate it because we can hear you. Okay, finally, you can use the flail mower also to get this brush chopped, or just pick it up and remove with the gator. You can spray copper, low rates of copper and apogee at this time, because you want to slow down the progress of the pathogen through the shoots. That's why apogee is going to help you, and also you don't want to promote the growth by cutting these branches. So apogee is going to help you keep that growth stunted, specifically of the shoots. And if you do this cutting for this low infection scenario rapidly, then you stand a chance of not having other continuing infections of the shoots throughout the season. And keep on scouting, and always cut these shoots on a cool and dry day. But if you have a large number of these clusters infected and strikes, about 25 to 30, that most likely means that you have had a big gap in your spray program. You likely didn't protect against fire blight during bloom, and you have allowed a severe infection to occur. So in these cases, the most important thing to use is apogee at a high rate, 12 ounces per 100 gallons, as soon as possible, so you can shut down the growth of the tree and also prevent the pathogen to invade through these shoots that are infected. So, and then continue applying copper at low rates, and also mix the copper with about 1 to 3 pounds of hydrated lime, so you can safeten it and allow slow release of copper in the future from the residue. So by applying apogee, you're slowing the migration of bacteria to the larger limbs. You're trying to prevent formation of cankers. And in general, by using copper, you're trying to prevent the spread of the infections on other shoots And then you have to go in and do the severe pruning on a cool, dry day so that you can get this inoculum out. You can scout, but if you have this many infections in the orchard, what's going to happen most likely is that you're going to promote by cutting more growth, and that's potentially going to create more susceptible tissue and more infections down the road. So if you do prune hard to get rid of all these infections, apogee is going to be your friend because it's going to help you stunt that growth that comes after the pruning. Okay, so what about midsummer? At any time after the infections are visible and the bloom is done, do not use streptomycin. You are risking potential development of resistance because now in the orchard, if you see the symptoms, each of these symptoms can have a lot of bacterial ooze there, and they're packed with bacteria. There's a lot of bacteria there. And if you expose them to streptomycin, you are basically increasing the chance for the mutations to occur. Or actually you are selecting them, increasing the pressure for the resistance to occur. So that's playing with fire. Don't use it. Don't use streptomycin later on in the season. So, and it's already done. The bacteria is inside of the flowers and shoots and wood. There's no way you can reach them with the streptomycin. So there's no point applying it. So there's danger plus, there's no point for you to reach the bacteria inside. What you want to do is we want to prevent the inoculum spread to those thousands and billions of bacteria that showed you they're present on the surface. So use copper before and after the pruning. Use lower rates because you want to avoid rusting on the fruit if you're a fresh fruit cultivar producer. Limit the, in general, you want to limit the amount of copper. So low rates, about 3, ounces of metallic copper per acre is enough. And you want to try to use, any formulation that you have, but Cuprofix, Badge is good, Coside is fine, Bordeaux mixture is good too. So you're trying to avoid the slow drying conditions, of course, to prevent rusting on the fruit. And then if you continue using low rates or high rates of apogee, that's going to help you, and especially use the low rates of apogee on trees that are younger than 5 years. The fully mature trees beyond 7 years use higher rates. And in general, the time when you should start using it is about 1 to 3 inch shoot growth, which will be late bloom or maybe petal fall, and then repeat it 14 to 21 days later. That's going to help you stunt the shoot growth and slow down the pathogen, movement through the shoot. And it's going to limit the new infections and make the shoots more resistant because it thickens the cell walls, preventing the bacterium to infect. In general, what you're trying to do with apogee is you're trying to bridge the time up until you get the terminal bud set of the shoot. When the terminal bud set, occurs, the shoot susceptibility goes rapidly down to fire blight. Okay, what about the continued pruning late in the summer? If you keep on scouting to see if you're going to get more shoot infections, that's actually the basis for you to decide when to cut. It can also be very difficult to determine when the terminal bud set occurs in different varieties. So if you have varieties, they have a longer time to reach the terminal bud set. Keep in mind that's definitely something you have to pay attention to. And in general, you prune, it can promote new shoot growth. So in general, you apply the copper before and after the pruning. Please mute yourself, okay? Please mute yourself if you are on this call. Okay, good. Okay. If you get the new shoot growth because of the pruning, it needs to be covered with copper, okay? You're trying to disinfect those newly developing shoots, and about 7 to 10 days interval is important using the low rates of copper up until you get the terminal bud set. And again, don't spray copper when you know you're going to have a slow drying, like from rain. And in general, always, try to don't do the hand thinning on a wet day. Try to find a good, cool, dry day when you do the hand thinning, and then after you're done with hand thinning, apply a low rate of copper. Okay, if you have a young orchard, they are prone to later blooming, so they're going to have a lot of red tail bloom. keep on, either defruiting those trees, try to apply the mix to get the flowers off or remove them mechanically. So you can use ActiGuard. It's a really good tool. Carrie Peters' data shows that it's very effective, by, spraying the cuts when you have young trees that are infected. Use the spray bottle and prune as soon as possible on a cool, dry day. Don't prune in the rain. But in general, on young trees, there's a high chance that pathogen has moved really, really down into the rootstock and it can kill the whole tree. But in general, if the pathogen, if you see the symptoms, into the leader of the tree, you can try to cut the leader and maybe regrow a shoot. But many times what is necessary to do is just remove and replant, because it's going to be likely that the pathogen is going to linger there and kill the whole tree eventually. If you have the early control effective, in generally, suckers should not be. You should just never get the infection on the suckers. So because if that happens, is you can get the pathogen easily get into the rootstock, and then if you have an M26 or M9, those rootstocks are going to die pretty quickly from fire blight. Don't use excessive nitrogen. That's going to promote more susceptible tissue to develop, and try to avoid irrigation at that time. Definitely sanitizing tool is going to help you to prevent the pathogen to spread further. And we don't have any data on whether the apogee can work on preventing infection of the suckers. We didn't do that research. We tried to get it funded. We never were able to do that. Finally, the hail. During the summer, if you get hail and you have had active infection in there and you removed the symptoms, there's a potential that you're going to get the wounds open on your bark and on your shoots. Sometimes if you don't see the wounds on the leaves, the hail can cause enough fruit injury for infection to occur. So, and if, by the time when in late summer, when hail maybe can occur, you're going to have a lot of other cankers that are oozing the bacterium out. So it's very important that this is the only time during the summer, if you had an active infection, that you can apply, the streptomycin along with Regulate to try to prevent a trauma blight on these trees. So try to do it up to 24 hours after the storm that had hail, applying streptomycin with Regulate. So keep in mind that this is something that is limited by the pre-harvest interval. So it says on the label for firewall that you should not apply it 50 days before harvest. So keep in mind that there's limits to this, and on pears it's about 30 days before harvest. So it's not like if you get a hail closer to harvest that you have to apply this material. No, you actually have to follow the label. Okay, finally, I'm going to close with a few more things that this RIMpro has inside incorporated. I'm showing you the monolinear model that is for stone fruit. This model is very interesting because it has the ability for you to choose whether you have, whether you have, a mid, late maturing or early maturing varieties of stone fruit. And then it shows you here the potential for the blossom blight caused by monolinia. So you can see here that it shows you that the flowers in the central graph, it shows you here that there's a susceptibility of flowers for the blossom blight infection. It's a very narrow window here, okay? We usually don't have issues in Virginia with blossom blight infections. But if you look at the coming closer to a harvest, you can see here that the susceptibility of the fruit becomes a problem, and that's where the most of the fungicides should be applied. In general, also, you can see that the model gives you potential for the latent fruit infections that can occur in the top graph here, the purple color. And also, it can show you whether there's a potential for infection depending on the actual weather conditions, which are all these orange peaks at the bottom that you can see. So it's a pretty interesting model. I haven't had a chance to use it, but in general, I wanted to get your attention to it, that you can actually have also something that can predict the infections for monolinia, and you can use it to apply your fungicides much more targetedly. I wanted to kind of show you like a very, I would say a lean and mean spray program for peaches. This is from Bill McIntosh. So, the peaches require, to a certain extent, less amount of fungicides than apples. in general, but you can see here that that in general you can use the Xyrem in fall, and also because that material can help you control the leaf curl. And if the leaf curl last year has not been a problem, you can use the copper in fall or Xyrem. And in general, but if the leaf curl was a problem, probably the fall application of Xyrem late fall is really important, and also before bud swell. The issue with applications is in early spring is you can have mud in your orchard, and you might not be able to get into the orchard. So many times the application in fall is preferred than the one in spring. Later on, when you go through the bloom and just get to petal fall and shock split, Bill recommends here using RIMe and as a good material to control and also initiate for scab and rusty spot. So really good material for the money. And then, first and second covers would be sulfur and RIMe, or you can, to control rusty spot, or, you can do Captain and Topsin. And also the third and the fourth cover, you can use sulfur, Captain, and Topsin, and then you can add some insecticides, as you can see here. So it's pretty cost-effective method to control the diseases. And specifically, I'm mostly concerned here about rusty spot and the powdery rusty spot and the brown rot. And finally, when you get to the pre-harvest period, now you have to start using materials that are going to be effective against monilinia, because the fruit are they're starting to mature, and they're much more prone to infection. So you have to keep on applying. About 3 weeks before harvest, you have to apply Maravon and Indar and add some insecticides so that you can control the infection of the brown rot, so that you don't have it on the fruit. So it's pretty economical model. Finally, RIMpro also offers you, within the same subscription, besides if you get access to Scab model and Fire Blight model, you can have also access to a plant stress model. It gives you the ability to use the weather data to run the model that tells you whether you can anticipate either drought or waterlogging, and drought from extreme temperatures. And this is important because looking at this, plant stress model can give you an insight whether you're going to get these conditions of drought that potentially would allow more bitter rot infection during the summer. So Colototricum causing bitter rot is a huge issue, and it especially is more devastating when you have very dry periods, of 85 Fahrenheit and above, like probably up to 95, which stresses the plant, and the plant is more, stressed because of the drought. It allows higher infections by the bitter rot. And looking at when the plant is stressed is also going to allow you to irrigate your trees ahead of these stress periods. So that also is within the model, models that you can access here. Finally, also the RIMpro has the model for sooty blotch that you can see here. So it will indicate to you a more accurate time when the first infection can occur. So it gives you at the bottom the indication of when the infection occurs, when the spores are germinating, and also when will you see the symptoms up there in the top graph. So you can see the visible mycelium as the dots, which are the gray and the black areas. So that is all included within this one subscription. And finally, they also have the Marstonina leaf blotch model incorporated there too, which also is probably good to use to determine when the first infection is going to occur. So these red lines can indicate to you that, and also it can give you information when the first symptoms are going to occur and be visible on the leaves. And with that, I'd like to thank you for your attention. I know this was a little bit long. This is my team. You can contact me with any questions on my email. And if you have any questions now, we can discuss them through this session. But if you have any other questions, you can email me at any time. And I would invite you to visit my lab's website and follow the blogs that you can get on this website. Thank you. So if there's any questions, let me know. You can unmute and we can talk more about it.
Hi, I got a question. With the if you're getting a weather station to use with RIMpro, is it critical to get one that has a leaf wetness sensor, or 'cause we've got like an existing one but it has no leaf wetness sensor on it? How critical is that as an input to the model?
Yeah, it is essential to use the leaf wetness sensor on the station, and both the Kestrel and the Rainwise. I think the Kestrel bought the Rainwise now. The two stations that are available on the NUA's website, basically that are compatible with the NUA network to be able to work with the NUA network, they actually have the leaf wetness sensors on them. So it is very important to have that, yes.
So we've had some issues getting accurate readings off of the Rainwise weather station that we have. Does it work with any other weather station, such
Yeah.
as Pearl or what was the one we talked about?
Yeah, so, well, the most important thing that you should do with your weather station is almost every second year you have to service it. And the Rainwise stations are known that they need to be sent back into the company so they can calibrate everything and then send it back to you. Usually this is what we recommend at the end of the year to the growers. And because of these, finicky issues, okay, with the Rainwise, there was another vendor that was introduced. So if you go to the NUA's website, now you can actually see that they use, actually, let me see, weather tools. They offer two different vendors of the stations you can purchase. So
Hey,
it's,
Sir Jan, it's John. Sorry, I had to hop in and out. Buy a weather station. There you go. So
yeah,
the other vendor is Onset Computer.
correct.
Onset.
Yeah. Thank
Yep.
you, John. I appreciate that.
Yep.
Yeah.
And, you know, to be blunt, I mean, you're right. Rainwise was bought by Kestrel, so I don't know what their policy currently is on recalibrating their stuff. So just keep
Yeah.
that in mind.
Yeah, it's a good point. In the past, before they were bought by Kestrel, we would always recommend the growers to go and send the machine to be recalibrated. So
Yeah.
now there's this other, onset that you can use. So you can just go and read through this. Everything is, available here on the on this web page. So I don't think actually this button here is working, but all the information about the two different things are just right here.
Down below it says buy a weather station. It has a link to Onset data loggers.
Yes.
There you go.
Okay. And does dew count as an infection period for fire blight?
Yes, it can. Absolutely, it does. And the model, the newest model has the ability to follow the dew, and that's why you need the leaf wetness sensor, because it's going to pick up the dew as well. And any form of water, dew, your spray water, or rain, is going to be an infection period. Yes. Okay. If you have any other questions, maybe you can put them in the chat. But you have my email that you have seen, and I'm going to post this video and the presentation on the same blog post. Actually, I'm going to make a new blog post. So if you have any questions, don't hesitate to contact me. If there's nothing else, I'd like to thank you one more time for your attention, and feel free to contact me in the future if something else comes up as a question. Is there anything else?
I have one more question.
Sure.
The Rainwise customer support that we've been using is very lackluster. Do you have another number possibly that you could email us?
You mean like a service or like a?
Yeah, so when we had trouble with our station, we called Rainwise and talked to Ryan, I think,
Sure.
and it wasn't very productive.
I
So I
see.
was wondering if there's anybody else at that company you can talk to.
Yeah, I had a couple of emails. I just put in the chat box right now all the numbers for, but I know that Eric Rollins is the person who I've communicated before in the past. Okay.
Mm-hmm.
I pasted the email of his there. I know that he would be the one who would help a lot. There was also another email that I can't find right now, but I'm pretty sure that sending an email to them would be the best course of action. I know sometimes they don't respond because they're busy, but I think that's the best shot I can tell you. So a lot of people decided to go with the onset because of that.
Okay. Thank you very much.
Sure. Lynn, did you have a question?
Yes, when you were explaining the timing on the spraying of the strep, when you have several days of infection all in a row,
Mhm.
it didn't make sense to me. Can you do it again, please?
Sure, absolutely. Let me open the presentation. Okay. So you see what happened about 3 or 4 years in the last 9 years. What I've seen is that you have these back-to-back infections, right? So many times, what people would ask us is, when do I apply streptomycin? Technically, the best time to apply would be on the May 2nd, okay, a day before the infection. However, when you apply on the May 2nd, you would not be able to cover for the infection of the May 5th. Okay, so I've been often caught recommending to people to do 2 applications of streptomycin, okay? And one that I would recommend would be the May 2nd and May 4th, okay? But then, you have a light rain on that day, it's wet. So then what I did is I basically went on and put in the date when the application of streptomycin were, okay? So in one case, I decided to apply the streptomycin on the May 3rd. So you have that ability to add that date even in the current model online. So you see, I applied one on May 3rd, and I was able to recalibrate the model, and it shows me that I was able to control the infection of the 4th and the 5th. But if I decided to apply my streptomycin on the May 4th, I would not be able to control the infection of the May 3rd. So what I'm basically just trying to say is, you have to play with the model to put in dates inside to see how model is going to recalibrate, so you should be able to cover yourself, to cover and prevent the infections in the flowers by looking at the way how the model is going to look like after you apply your streptomycin.
Okay, so in the past, we've always debated as to, are you going to do spray when the risk is high or when it actually says there's an infection? Because sometimes I worry that, maybe we should have looked at the model 2 hours later and gotten a different reading and gone from orange to red. And it's so chancy.
Yeah, I was going to say, I know that's the confusion that this model creates for a lot of people, okay? When the infection potential value is not the infection. It just tells you that you on the flowers have enough bacteria to cause the infection if the rain occurs. So if you have this number at 157, let's say on May 2nd, right? That just means that, yes, there's bacteria there, they're growing on the flowers, but they're not going to infect until you provide the water to them. So the model then turns into the infection in red if the rain is predicted or due. Okay, or maybe let's say you want to apply your fungicide, right? And the model is telling you, oh, there's a high risk. Okay, well, I know this is high risk, but I'm going to spray, I'm going to provide the water, so I should add streptomycin into my tank, right? That is very logical and should be done. But the high infection, like orange field, does not mean necessarily you're going to have infection. It just means that everything is ready except rain or the water there. So that's why we usually go and primarily look at that the EIP is above 100. This field turns into the red and tells us that there's a prediction of infection. And then we decide based on that one when we're going to apply the materials. So, you know, this is based on the experience of many pathologists in the past. So my concern primarily is not so much about these colors. We try to explain that at the extension meetings a lot, and many times people then eventually get it. But my biggest concern is the like 5 or 6 infection days that occur during bloom that you know, really don't know how to approach managing that. Because if you have several infection periods like I'm showing you here, on each of these days you're going to get a new group of flowers opened. And these flowers that open on the May 4th or the 5th, they're going to be not protected, because if you applied the material on the May 2nd or the May 3rd, right? Because the streptomycin has not been able to enter inside of the flowers. So that's the problem that we have with the climate change, because it allows these blocks of time with very extreme favorable conditions for fire blight. And we've done many different tests. When we would do an application of streptomycin at the first infection and the last, we would still get infection in the middle. So it's just unfortunate that this happens, and it's much more mind-boggling for growers to fathom, and you know, to figure out how to protect against it.
So in the spray if you have an infection period, you were recommending both streptomycin and oxytetracycline. So is it better to use one, and then if we're going to have another infection in a few days, use the other one? Or do you want to use both of them because of resistance? And if you use both of them, how long do we get to use both of them in the same spray?
So I personally would do both of them. Okay, that's based on the you can use streptomycin alone. Okay, I haven't had any, for at least for Virginia, where I move more frequently, I haven't had any people complain to me about the efficacy of streptomycin slipping. But I do it because I see a lot of West Coast growers always mix Firewall and Fireline together, you know, and then apply Regulate. But I guess in the mid-Atlantic, I haven't heard any issues for Maryland, you know, for resistance. Maybe I didn't look, but I'll tell you that streptomycin, you know, is really effective. If you apply it with Regulate, you're allowing that penetration. So I just add the Firewall there as another, like, our anti-resistance management strategy. Okay, that's what I would do. And I would do that mix of both antibiotics first time, and also mix of both antibiotics on the second time. So that's kind of like the thing that what I would do. But if you just do Firewall with Regulate, it should be equally effective alone on that first day or in the second spray as well. The issue becomes the Regulate. Okay, Regulate is the material that if you apply more than 2 times Regulate with streptomycin, you can get the leaf yellowing. Some people don't like that. And many times we just tell them, just apply for the first infection period, apply your streptomycin with Regulate. And then for the subsequent applications, if you are concerned about the leaf yellowing, back off on adding Regulate. But many times we see that the leaf yellowing actually is an indicator that we had a good absorption of the streptomycin inside of the leaf tissue. And trees really, you know, usually grow out of it pretty well.
Okay, thank you.
Sure, anytime. Any other questions or comments? Okay. So if I don't see anybody else discussing and commenting, I would thank you for your attention. Okay, I see somebody in the chat. Okay, anytime, Steve. I appreciate that. Stay in touch, and thank you so much for attending, and I'll talk to you later.